Hello everyone. Today we are going to talk about non-disjunction and its effects on individuals who carry these genotypes. So a non-disjunction is simply a case where during anaphase, either anaphase 1 or 2, the chromatids are not separated. And because the chromatids don't get separated, this results in gametes with extra or less chromatids or chromosomes than there should be. If you, in a gamete where there is an extra chromosome, we call this a trisomy. So here you can see an example where there is 2 copies of one of the chromosomes from one parent or two, grandparent, I guess, chromosomes and one extra one. And in the other case, there is one missing, which is a monosomy. This occurs when chromosomes or chromatids do not separate, and if these are the gametes that are involved in fertilization, then the embryo will have extra or fewer chromosomes. There's a rare survival chance in the case where chromosomes are missing or there are extra chromosomes because this is a careful, balanced program in the cell development where if the addition of an extra chromosome often will result in too much of certain proteins being formed, which will interfere with the way the cell develops. And if there are less chromosomes, then there's not enough protein, and that will also cause problems. And so here you can see some ways that this could happen, either in meiosis 1 or meiosis 2. In meiosis 1, you end up with 4 gametes, all of them having an unusual number of chromosomes. In the case of it happening in meiosis 2, then only half of the gametes are affected. These are often diagnosed using something called a karyotype. This is a lab technique where the cells are frozen in an intermediate stage of the mitosis, and when the chromosomes are condensed and separated from each other. And so by freezing out the cells during that point and then also rupturing the cells, they can spread the chromosomes out a little bit, and they will take a large picture of all of these broken cells, and they can look at the different locations and essentially analyze each cell and that they can find and try to cut out. Like in the olden days, they would just actually blow this up on a giant piece of paper, and they'd get a pair of scissors and they would try to cut out every single chromosome and arrange them on arrange them together and look for the ones that were similar in length. They can dye them so that they can tell that they're homologous by the pattern of black bands on the chromosomes. And as you can see on the right here, this is a karyotype here. And they can take pictures, and they essentially do this for enough cells that there is at least one usually that works out that has all the chromosomes clearly visible, and then they can compare that to the others. And so here on the right we see one karyotype where we have all 22 chromosome pairs and the X and the Y chromosome over here. And so this is a typical cell for an individual where the meiosis has gone in the typical way. And in the case of in a case of a non-disjunction, there are a few examples. One of those examples can be an autosomal non-disjunction. So that would be the case where one of these 22 chromosomes are affected, and that would indicate that we'd have an extra or one less chromosome than we expect. This is often a fatal condition for the zygote. It can essentially cause an autoabortion because, or a miscarriage, I should say, because there's so many genes located on those chromosomes. And so this can result in the cell missing essential structures, or it can result in the cell having too much of some protein or whatever, using up important materials. Now, Down syndrome is an example of a trisomy that is in trisomy 21, so that's this chromosome right here. And so that is an additional chromosome. You can see it here. One extra chromosome 21. In fact, sometimes it's not even a full chromosome. Occasionally, in a non-disjunction, it's just a piece of the chromosome that gets carried over. But this has a huge effect on the individual because there are some variations in how much of that third chromosome they get and and differences in the way development occurs through the development process. Individuals living with Down syndrome have a variety of abilities and or a variety of challenges that they live with. Some live with very few few challenges or exceptionalities. Others live with quite a few. My own brother was born with Down syndrome. Was born with he does face many intellectual disability challenges and heart problems, heart surgeries throughout his childhood to fix some structures that didn't develop in a way that could support him, and characteristic facial features and changes in stature. So this creates a number of chromosomes that we actually want to try to avoid the word abnormal if we can. I am trying to develop you'll see in the next section that I'm trying to develop the language, but we can just call this a number of chromosome. We can just call this a chromosome number. Get rid of that normal part. We don't want to compare two individuals and create them as two categories. Everybody is an individual, even if they have to live with challenges. And so this creates a chromosome number of 47 and a variety of effects. It affects 1 in 800 children in Canada. It is categorized as a syndrome, and if you're not aware what a syndrome is a condition characterized by a set of associated symptoms, and the symptoms may be are associated, so they aren't guaranteed. They might have those symptoms, they might not have those symptoms, but all of those symptoms, but this sort of set of symptoms are all associated with the same instigating factor. So in this case, the trisomy. So having this trisomy can impact this. It typically results in a set of symptoms, of which they might be more or less on that scale. So in the other possible non-disjunction, the more common one in a way, I guess, is the non-disjunction of sex chromosomes. This is a cell. So that means the non-disjunction of particularly the X chromosome, and this is a more common non-disjunction because the X chromosome, we recall, has that bar body effect. So X chromosomes already have there's already a mechanism in the cell for turning off X chromosomes, and with the non-disjunction of extra X chromosomes, those extra X chromosomes can, in many of the cells, just form bar bodies and not interfere with the development of that cell. And so these cells are now able to, at the very least, form up into to develop past the zygote stage and into the individual stage, or into the fetus, and then onto being born. So if the egg cells fuse with normal sperm cells and sex chromosomes with the sex chromosome zygote that have these non-disjunctions, typically we're talking about non-disjunction happening in the egg cells. Since the egg cells, I'm not sure exactly the reason. I suspect that the sperm cells, maybe there are so many sperm produced in a given time that the success rate of non-disjunctioned sperm cells is low, but I'm not actually certain about that. But we almost always discuss the non-disjunction of sex chromosomes happening in female cells, and I just put it together. It's because female cells are the ones with 2 X chromosomes, of course. We can't have much of a non-disjunction in the male cells because they only have an X and a Y chromosome. Well, I guess there's still potential, but since non-disjunctions typically happen when the 2 chromosomes are side by side, it's only in the female chromosome, that the female egg cells, that this is more common. Okay, that makes more sense to me. So the first syndrome is known as Turner syndrome, and as I said, I'm trying to update the language so you don't have to replace your own language, but I am trying to update it to be more appropriate. So this, the Turner syndrome is when the egg cell actually has no X chromosome, but a sperm cell comes with a chromosome. So you can see it up here, drawn out there. The sperm cell bringing an X chromosome, the female egg cell having no chromosome, here placed as a 0 or as an O. And so this will cause a female phenotype. So the genetic development, or the genetics will develop, or the phenotype development of a female genitalia and things like that. But those individuals may end up living with reduced height development. They might have what is known as webbing in the skin of the neck. This is sort of additional skin that grows in the neck area. There are examples of cognitive disabilities for these individuals, as well as underdevelopment of the ovaries and the breast tissue. They may also live with a lack of body hair and possible fertility issues. Fetuses with this genotype do have a 1 in 10 chance of being miscarried. So it is not a particularly successful genotype for the fetus. And you can see here, this is the example of the karyotype, where you can see the missing chromosome over here. The next example is this is just my writing, is the XXX. This is where there are an additional X chromosome from the female egg cell. And so in this case, the individual is born with a female phenotype, but they have they have very little impact on the physical development. Physically, they can develop without differences from others born with the XX genotype. But they may have some impact on the neurological development, so they might live with a cognitive disability, for example. But in the end, it does not seem to have much of an impact on fertility. They still have both X chromosomes, so their egg cells will all have X chromosomes in them as well. And the genetic change does not seem to be transferred to offspring. So I'm not sure exactly what happens there. If during meiosis, the eggs with the 2 X chromosomes just have a lower chance of being carried through to the egg stage. But that's what the information says. And I did have here living with cognitive disabilities, which is the same as what I said there about neurological development. Oh well. Anyways, Y0 is a monosomy, but that never occurs. So this would be an egg cell that has no chromosome, but no X chromosome, and those egg cells cannot be fertilized even if the sperm cell contains a Y chromosome. It's just not a possible genotype. You need at least 1 X chromosome to survive. So that's a non-example in a way. And then Klinefelter syndrome is the XXY genotype. So that is where the egg cell contributes 2 X chromosomes, but the sperm cell contributes 1 Y chromosome. This results in phenotypical male sex development, so sex organs of male sex organs. And then these individuals may live with changes in the development of their testes. They might have fertility challenges. There are the possibility of enlargement of the breast tissue and a lack of body hair or changes in the body hair placement at puberty. And they might also have cognitive disabilities or might live with cognitive disabilities. So those are some examples. I'm going to post some videos to sort of give us some viewpoints into the life of individuals who live with some of these non-disjunctions. These are real individuals and real people. They're not just clinical studies or words on a page. So hopefully that will give you some empathy and some ideas of the challenges that are inherent in this type of genetic change. And so with that, I'm going to say thank you and hope you have a great day.